Hello, my name is Petrosyan, and today I'm going to be discussing the extracellular matrix in renal development and cancer. Just a brief background about the extracellular matrix. The extracellular matrix is highly complex. It is composed of both physical signals, such as fibronectin, laminin, and collagens, and it is also made up of soluble signals, such as growth factors, cytokines, and chemokines. We also have integrants, which are transmembrane proteins that directly link the actin cytoskeleton with the extracellular matrix to transmit signals bidirectionally between the extracellular matrix and the cytoplasmic domain. And this is to promote cell adhesion and recognition during embryogenesis, homeostasis, tissue repair, immune response, and metastatic diffusion of tumor cells. And this is an important concept because in cancer, the extracellular matrix actually plays a huge role. For example, in breast cancer, in normal mammary glands, we have the basement membrane made up of laminins and collagen 4, and we also have the interstitial extracellular matrix composed of fibril collagens such as 1, 3, and 4, also fibronectin. However, when we have breast cancer, we actually have a disruption of this basement membrane, and we could have a decreased deposition of collagen 4 and laminin 111. Also fibronectin. However, when we have breast cancer, we actually have a disruption of this basement membrane, and we could have a decreased deposition of collagen 4.
Yes, can you hear me?
Yes, I'm being repeated. Also fibronectin. However, when we have breast cancer,
okay, interesting.
Disruption of this basement membrane, it is really weird. Deposition of collagen.
Yeah, let's go ahead and have you exit the studio and then re-enter.
Hello?
Yeah, let's go ahead and have you exit the studio and then re-enter.
Okay. Tyler, can you hear me?
Yes, can you hear me?
Yes, I might have opened some other web browser, and I think it kept recording. I don't know, it was playing back whatever I was. Can we start all over?
Yes, it seems to have been the case. So what slide were you on?
I was on slide number 2.
Okay. If you'd like, we can go ahead and just start fresh from the beginning.
Yes, I would prefer we start from fresh.
Okay, let's go ahead and do that. Let me go ahead and make a quick editing note, and then we can get started again.
Okay.
Okay, all set. So just go ahead and give it a few seconds of silence, and then you can start again.
Hello, my name is Petrosyan, and today I will be discussing the extracellular matrix in renal development and cancer. A brief background about the extracellular matrix. The extracellular matrix is composed of physical signals such as fibronectin, laminin, and collagen, and it's also composed of soluble signals such as growth factors, cytokines, and chemokines. We also have integrants, which are transmembrane proteins that link the actin cytoskeleton with the extracellular matrix to transmit signals bidirectionally between the extracellular matrix and cytoplasmic domains. And they do this to promote cell adhesion and recognition during development, homeostasis, and disease processes. In particular, the extracellular matrix also plays a role in cancer. For example, in normal mammary glands, we have the basement membrane, which is composed of laminins, collagen 4, and also the interstitial extracellular matrix, which is composed of fibril collagens such as type 1, 3, and 4, and fibronectin. However, when we have cancer, we actually have a disruption of this basement membrane, and we see a decrease in collagen 4 and laminin 111, and while we see an increase in ECM remodeling MMPs. We also see a stiffer interstitial extracellular matrix with more deposition of fibril collagens 1, 3, and 4, and fibronectin. So this brings into an important concept, the fact that we need to be able to characterize the extracellular matrix of cancer in order to develop newer innovative treatments specifically designed to target specific parts of the cancer, such as cancer stem cells, and in a way to reduce the risk of reoccurrence and metastasis. I will also be discussing the kidney. And here's a brief background about the kidney. The kidney is highly complex and it has multiple functions. The functional unit of the kidney is called the nephron. And one of the functions of the kidney is to filter our blood. And blood filtration occurs within the glomerulus as shown by the pointed arrow. We also have a beautiful schematic image of the kidney extracellular matrix, in particular the nephron. And we could see that depending on the location of the nephron, you have different deposition of ECM proteins. For example, in the glomerulus, the glomerular basement membrane has to have the deposition of collagen 4, alpha 3, 4, and 5. However, when we have mutation within any of these chains, we have something called Alport syndrome. And unfortunately, patients with Alport syndrome, they have decreased kidney function and eventually will need a transplant. So this brings an important point that we do have a characterization of the ECM of the adult nephron in particular. However, little is known about the extracellular matrix in renal development. And that's what we wanted to do, and that's to characterize the extracellular matrix in fetal development. And here we have a histology of fetal kidney at 10, 16, and 20 weeks. An H&E and a picoserous red, where the red denotes the deposition of collagens. And we could see that over time, there are changes that occur from 10 weeks to 20 weeks, from A to C. We see that, in fact, we have a reduction of the nephrogenic zone. We see presence of more developed structures of the nephron, such as glomeruli, vasculature, and tubules. We also see increased lobes, and we also see distinguishable architectural structures of the kidney, such as the renal pelvis, medulla, and the cortex. And looking at the picoserous red in D, E, and F, we could see from D to F that we have more complex and organized deposition of ECM. And this is quite important for the function of the kidney. Moving forward, we also wanted to look at the particular fibrous proteins, such as collagens, fibronectin, and laminin alpha 5. And we've done immunofluorescence staining at 10 weeks, shown in A, C, E, G, and I, K, M, and O, and a 20 weeks staining shown in the rest. And what we could see that over time, we do see changes in the distribution of the extracellular matrix expressed. For example, for fibronectin in A and B, at 10 weeks, we see the deposition of fibronectin throughout the fetal kidney. However, at 20 weeks, we can see more specification, mostly in the cortex of the kidney. We also see the same thing for collagen 16, shown in K and L, where at 10 weeks in K, we see distribution of collagen 16 throughout the fetal kidney. However, in L, at 20 weeks, we really see a specification mostly within the renal cortex, specifically the nephrogenic zone. And we see also for collagen 4, alpha 1 and 2, shown in G and H, the deposition initially at G in developing structures, but at H, it seems to specify mostly within the glomerulus. So we believe this enhanced organization expression of ECM might actually be dictating cell differentiation versus stemness. So what we wanted to do is look into the nephron progenitors. The nephron progenitors actually give rise to the full mature nephron. They're identifiable by transcription factors SIX2 and CITED1, shown in the red arrow. They're found within the kappa mesenchyme, and they interact with the ureteric bud. And over time, they are induced to differentiate. They make developing structures such as renal vessel, comma-shaped, S-shaped body, and they eventually form the full mature nephron. And most of the studies done on these progenitor population have been more focused on intercellular events, more molecular studies. And what we see, what we wanted to do was actually characterize the extracellular matrix and hope to identify ways that the extracellular matrix might promote their stemness rather than their differentiation and also their motility survival. And so our aim was to characterize the extracellular matrix within these nephron progenitors in normal human developing kidney, and also identify how extracellular matrix might be promoting their self-renewal versus differentiation. And we wanted to use the information that we obtained from this to actually apply it in vitro for us to be able to culture these cells for an extended period of time. So here's immunofluorescence staining of a 10-week fetal kidney that identifies these nephron progenitors, SIX2 identified by red, CITED1 in green, and DAPI showing nuclear stainings. And we could see that the expression of these markers are in the nephrogenic zone, in the outer layer and inner lobes of the kidney. And we could see that over time, initially, around the cap mesenchyme, they have strong expression of SIX2 and CITED1, but once they make the developing structures such as the renal vessel, C-shape, and common-shaped body, they begin to lose the expression of CITED1 and SIX2, and once they make the full mature nephron, they do not have the expression of these markers. So in normal development, nephron progenitors should disappear before or soon after birth. However, in some cases, this progenitor population are left behind. And this gives rise to Wilms tumor. Wilms tumor is a pediatric cancer, also known as nephroblastoma, and it accounts for 95% of renal malignancies in children. It's quite rare. It occurs 1 in 10,000 to 1 in 8,000 children. And Wilms tumor is believed to originate from abnormal renal development and tends to histologically resemble fetal developmental stages, not found in postnatal mature functional kidneys. So our goal was to characterize the extracellular matrix within normal developed nephron progenitors and Wilms tumor nephron progenitors that also express SIX2 and CITED1. And here's an immunofluorescence staining of a 10-week human fetal kidney showing the expression of SIX2 CITED1 found in their proper location in the cap mesenchyme, while on the right, we have a stage 3 Wilms tumor of a 6-month-old patient. And we could see, again, the deposition of this progenitor population that should not be present. Moving forward to have a bigger grasp on the extent of these progenitor population, it varies from tumor to tumor, but in particular, we see an extensive expression of SIX2 CITED1 within the Wilms tumor, and not in a proper organized manner, as we saw within the fetal kidney. In fact, most of the expression of SIX2 CITED1 is found within the blastema. And these are the most undifferentiated portion of the tumor. However, once the tumor attempts to differentiate and create abortive structures such as tubules and glomeruli, they lose the expression of SIX2 CITED1, and also the stroma does not have a strong expression of these markers. So next, what we wanted to do, just to grasp a general concept, was to look at the extracellular matrix composition of the Wilms tumor. First, we wanted to look at the histology, and we did, again, H&E and picoserous red staining. And we could see that, unlike the human fetal kidney, it is a disorganized mesh of cells. And in particular, this tumor had a lot of blastema. That is why it has a lot of these uncharacterized structures. And moving forward, we wanted to look at the extracellular matrix proteins that we have looked for the human fetal kidney. And what we could tell is, yes, it is a disorganized expression of markers. However, we distinguished that the strong expression of laminin alpha-5, collagen 16, and to some extent collagen 21 and collagen 18 is mostly found within the blastemal component, while expression of fibronectin, collagen 1, 3, and 4, alpha 1 and 2, is found more in the tumor interstitium. So next, we wanted to look at actually the nephrogenic progenitors, the nephron progenitors, their niche. So we looked at normal human fetal kidney, which is identified A through H, and also Wilms tumor blastema, I2P. And we could see the ureteric bud beautifully outlined by collagen 18 and collagen 1, shown in E and B. and the outer portion where we have the nephron progenitors. And in the blastemal component is the most undifferentiated component, so it's a mass of cells. And what we could identify is that indeed the human fetal kidney nephron progenitors and the Wilms tumor nephron progenitors share similar extracellular matrix characteristics. They express collagen 16, identifiable in F and N, and laminin alpha 5, identifiable in H and P, and to some extent, collagen 18, identifiable in E and M. Well, we see that the Wilms tumor nephron progenitor appear to have less expression of collagen 21, identified in G in human fetal kidney and O in the Wilms tumor. So this gave us a little bit of information. Next, we wanted to know even further. So we wanted to actually do single cell analysis on these progenitor population because we know heterogeneity does exist within these population. So what we had done previously, we were able to isolate these progenitor populations based on the transcription factor SIX2+CITED1, and we did this by using SmartFill technology, RNA probes. And we've published this data before, and we wanted to do the same thing with the Wilms tumor and see if we could also obtain Wilms tumor nephron progenitor population. And we were indeed able to do the same. And one important thing to note is in the human fetal kidney, the percentage of SIX2+CITED1 was pretty consistent. However, in the Wilms tumor, depending on the tumor type and stage, we actually had differences in expression of SIX2+CITED1. This particular tumor, which were predominantly more blastema, had actually 51% expression of SIX2+CITED1. Next, we just wanted to look in bulk if we see a difference between the human fetal kidney and the tumors. So we used 3 different tumors, Wilms tumor number 3, which is unfavorable, Wilms tumor number 4, which is favorable, and Wilms tumor number 5, which was classified as favorable. However, it was a stage 4 and the patient received treatment. So these cells have received chemotherapy treatment. And we wanted to compare it to human fetal kidney in our previous publication, which were around 17 weeks of gestation. As we could see that the human fetal kidney more clustered together, and they're very similar to each other, while the Wilms tumor, there seems to be variation between tumor to tumor. And we know this does exist. And next, we wanted to just look closer into the extracellular matrix, and we looked at collagens. And we could see that the Wilms tumors tend to cluster more together, while the human fetal kidney clustered together, and they share similarities between one another. While laminin, it seemed like they actually expressed similar laminins. In particular, they expressed a lot of laminin alpha 5. We also wanted to look at integrins. And again, the Wilms tumor appeared to cluster more together, and human fetal kidneys cluster more together. But one important thing to note is that there was a statistically significant difference in the expression of integrin beta 1. In fact, integrin beta 1 appeared to be highly expressed within the human fetal kidney compared to the Wilms tumor. So we thought this was interesting because there's a lot of information on integrin beta 1 and its importance in development, maintaining stem cell stemness, and also promoting cancer. So we wanted to confirm this. So we did immunofluorescence stainings of human fetal kidneys shown on the left panel for integrin beta 1 and SIX2 shown in red, and integrin beta 1 and CITED1 shown in green. And we compared it to the human fetal kidney for the same stainings, and we could see that indeed there seems to be higher expression of integrin beta 1 where we find these progenitor cells compared to the tumor. In fact, there seems to be a higher expression of integrin beta 1 by the ureteric bud, also by Wilms tumor developing structures shown on the right below, where we have tubular structure and they are strongly expressing integrin beta 1. So we confirmed that indeed human fetal kidney nephron progenitor show stronger expression of integrin beta 1 compared to the Wilms tumor. So this is interesting. Next, we wanted to actually look at it at a single cell level, and we wanted to identify if this was also the case. So what we did is we isolated Wilms tumor nephron progenitors, human fetal kidney nephron progenitors, and we did single cell analysis on them. And then we merged the data, and we wanted to identify clusters shared solely by Wilms tumor nephron progenitors and only by hemophilia kidney nephron progenitors, and some clusters shared together. And what we identified was, indeed, there was 2 clusters that were shared by human fetal kidney and Wilms tumor nephron progenitors, which were clusters number 2 and 7. But we were able to identify 6 clusters that were mostly expressed by Wilms tumor nephron progenitors. We also identified 4 clusters that were solely expressed by human fetal kidney nephron progenitors. So next we wanted to test if indeed the expression of collagens, laminins, and integrins within the clusters. Interestingly enough, we identified that indeed the clusters identified Wilms tumor nephron progenitors predominantly, shown in red and in red boxes, tend to cluster together. And they had much lower expression of collagens and integrins, specifically integrin beta-1, shown by the arrow. While clusters mostly expressed by human fetal kidney nephron progenitors appear to have stronger expression of extracellular matrix proteins. And clusters shared by both seem to have stronger expression compared to Wilms tumor nephron progenitors only clusters. But one of the clusters, so they share 2 clusters, one of the clusters did in fact have expression of integrin beta-1, but the other cluster did not. So in general, we also see that the shared clusters had higher expression of collagen 16, shown in arrow, and also collagen 6. So in general, what we could take from this is that they all express laminin alpha 5, shown in the arrow from the middle to some extent, while the Wilms tumor nephron progenitors appear to have much less expression of integrin beta-1. Next, we wanted to test if this low expression of integrin beta-1 and expression of laminin alpha 5 might be important in maintaining their self-renewal rather than because these cells are left behind and they're unable to differentiate and they keep maintaining the expression of SIX2+CITED1. And we wanted to use laminin 511 for laminin alpha 5. And we know that integrin beta-1 and laminin 511 do interact together. So what we did is in vitro cultures of normal nephron progenitors, and we tested out different culture conditions. In particular, we tested for tissue culture without any cleaving, and the cells do not like this, and they tend to not be able to attach. We tested Metrigel, which is the standard currently to culture these cells on Metrigel, and collagen 1, fibronectin, collagen 16, and laminin 511. And we tested through full analysis for the expression of SIX2 and CITED1. After 5 days, what we see that laminin 511, when the cells were cultured on laminin 511, they seem to maintain the expression of SIX2+CITED1 significantly compared to all the other culture conditions. And next, we wanted to see what if we block integrin beta-1 using anti-integrin beta-1 antibodies. Would this change the expression of SIX2+CITED1? And indeed, with flow analysis, we showed that in standard case using the Metrigel, we have a little bit above 20% maintenance after 28 days of culture. In laminin, a little bit more, while when we block integrin beta-1 and culture the cells on laminin 511, we are able to maintain their expression of SIX2+CITED1 significantly up to almost 80% in some cases. And this correlates to the Wilms tumor nephron progenitors cultured on just laminin, so they are unable to differentiate well, so they maintain their expression of SIX2+CITED1. So they're very similar. This brings us to a summary. Basically, what we find that Wilms tumor nephron progenitors and human fetal kidney nephron progenitors express similar ACM markers such as collagen 16, collagen 21, and laminin alpha 5. And this may be important to regulate their self-renewal rather than their differentiation. We also see with the single-cell RNA-seq data that clusters mostly expressed by human fetal kidney nephron progenitors have higher expression of collagens, laminins, integrins compared to clusters mostly expressed by Wilms tumor nephron progenitors. We also see that clusters shared by Wilms tumor and human fetal kidney nephron progenitors show stronger expression of collagen 16 and collagen alpha 6, as collagen 6 alpha 1. And collagen 6 alpha 1 is known to have a broad range of cytoprotective effects, and it has a role in maintaining stemness and also in cancer. We also see that Wilms tumor nephron progenitors and hemophilia nephron progenitors both express laminin 501 and collagen 16 constantly, and cluster predominantly expressed by Wilms tumor nephron progenitors show very low expression of integrin beta 1. So we can promote, we find that we can promote in vitro hemophilia nephron progenitor self-renewal versus their differentiation by mimicking the Wilms tumor nephron progenitor extracellular matrix niche by using in vitro coating of laminin 501 and blockage of integrin beta 1. And final thoughts are that we should consider that the role of the ECM extends beyond mechanics, and the local ECM environment is highly cell-specific. By studying in cancer stem cell biology and by studying and understanding and characterizing the extracellular matrix in cancer, we could actually find ways to provide more novel therapeutic targets for stem cells and understand why they stay in the self-renewal state rather than they differentiate. And we could do this by looking at the hemophilia kidney nephron progenitors and comparing to the Wilms tumor nephron progenitors. So currently, we are lucky to have new tools and new technologies constantly. We could characterize the extracellular matrix in different ways, such as quantitative proteomics. We could do decellularization. And we also have large data sets now that we could rely on and go into and identify what the extracellular matrix composition should be for each organ. So with that in mind, I would like to thank my PI, Dr. Labroots, also Dr. Daisako, who's a co-PI on this project, and my also Dr. Agulari and Viliani, who also worked with me on this project. And of course, we are the GoFar Foundation, and without the GoFar Foundation, we wouldn't be where we are, and our foundation, and also all the awards that we have received in the past. With that, thank you. Okay, I'm done.
Okay, awesome job. Well, we're all set. Pretty much from here. We'll just do some quick editing on the front and back ends and get your presentation ready for the event in a few weeks.
Okay, great. Was I talking really quickly? I tend to talk pretty fast sometimes.
No, I didn't think so. I thought that you communicated smoothly and clearly. Okay,
perfect. Thank you.
Awesome. Well, any questions for me before we wrap up here?
No. Well, one thing, would I be able to see the final version before you send it out? Or
definitely, I can send out a preview link once we've completed the editing.
Okay, great. Thank you.
Of course. Well, if any questions come up, feel free to reach out. But if not, I will send you that preview link once it's ready.
Okay, awesome. Thank you, Tyler.
Thank you.
Have a great weekend.
You too. Bye-bye.
Bye-bye.